This begins our series on the nature of genes. So this is your area of study 2 within your Unit 2. We're working from Chapter 10 in your Heinemann textbook for this. Your study design dot points that you'll be working on within this entire PowerPoint, but it will be broken up into a couple of different videos, is your distinction between a genome, gene, and allele. Your genome as the sum of a total of an organism's DNA, measured in the number of those base pairs contained in a haploid set of chromosomes, and the role of genomic research since the Human Genome Project, with reference to the sequencing of the genes of many organisms, comparing relatedness between species, determining gene function and genomic applications for the early detection and diagnosis of human diseases. So the first one we want to look at is DNA. When we're talking about DNA, we need to understand the structure to start off with. This is going to then be an introduction into our genome and what our genome looks like. So when we talk about offspring of individuals, and we talk about meiosis and mitosis, we're talking about the replication and the passing on of DNA when they go into their daughter cells. This DNA codes for genes. To understand these terms, we need to look at the structure of DNA. All cells get their genetic material from DNA or deoxyribonucleic acids. This information is hereditary and directs all of the cell's activities and is passed on from generation to generation through the prophase 1 section of meiosis. So remember, that's the crossing over part. So that crossing over creates our genetic combinations that are then going to be passed on from that gamete that it creates into the fertilized egg, which is our passing on of our hereditary traits. So I would like you to put DNA as deoxyribonucleic acid and hereditary as 2 of your cue cards. The third cue card I'd like you to write is DNA having regions known as genes that contain different genetic information that determines what you look like and how your body functions. So if you want a couple more pictures of this, you can look up 10.1 in your textbook. But we need to look at DNA as a large macromolecule, which is made up from a few different chemical building blocks. It's made up of a phosphate, this light yellow down the bottom, a sugar, which is called deoxyribose, and a single nucleotide. Now up here, there's a picture of 4 nucleotides. We have adenine, thymine, guanine, and cytosine. And these are the 4 bases, the only 4 bases that our DNA has. RNA has a base called uracil, which is also a pyrimidine, but we'll go on to that a little bit more in just a minute. So when we look at a nucleotide, we're looking at the phosphate group, the sugar, and the base all together. And this shows a single link on one side of your double helix. Whereas we go back to here, these are some of our parts. One thing I'd also really like you to know is the difference between purines and pyrimidines within the bases. So on the left-hand side, you've got purines, which are double ring, and they're adenine and guanine. And then you've got pyrimidines on the right-hand side, which are thymine and cytosine. So purines and pyrimidines. These could be some good words to have on your cue cards as well. So phosphate, deoxyribose or your sugar, and your nucleotides. Then on the other side, you could have nucleotides can be purines or pyrimidines, and they are made up of adenine, thymine, guanine, and cytosine. Or you could have those as some separate cue cards as well. So purine could be on a cue card, and then on the back you could say adenine and guanine are both purines. They have 2 rings. And then you can have pyrimidine as a card and say that thymine, cytosine, and uracil are pyrimidines. So when we look at that as a whole, what it does is it shows how we can create our nucleotide and our DNA as a double-stranded helix, which is something we'll go on to in just a second. So when we're looking at this figure on the right-hand side, D, this figure is showing a single polynucleotide chain link. Then the second dot point says the carbon atoms in the sugar are numbered 1 to 5. So when we look at these, we start with our right-hand side, the one that connects to our base. Wherever you can see kind of a join, there's a carbon there. Whereas see here how it's got the O in there, that's because there's an oxygen there linking it. So in here as well, we've got C, 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 but then they've got ends where there aren't carbons. And this one here is carbons here. So when we're looking for the carbons in the sugar, we need to be looking for these little points. So there's carbon number 1 here, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. And I'd really like you to label these 1, 2, 3, 4, and 5, because then we talk about DNA running 5' to 3', or 3' to 5', depending on which order we end up putting it in. So we'll come back to that in a minute. So you need to make sure, maybe you want to draw this. You don't have to draw the base. You could just write base attached to it, or you could give an example of any of the 4 bases, and we'll work with just the 4 for now. So this base would be a purine because it's got 2 rings. The only way that a nucleotide or adenosine monophosphate, which is a single one, is distinguished is by the base that it holds. The rest of the sugar and the phosphate group are the exact same. It's just where this nitrogen attaches to a different base that makes it different. Each nucleotide is joined to the next with a covalent phosphodiester bond. So we have hydrogen bonds that run from this side. So this one will have 2 links, but then we have a phosphodiester bond. So we have our phosphate group running out the back side, and these, our sugar and our phosphate, create the backbone. And these are smaller molecules compared to our nucleotide base. So something you might like to draw is this one here, the sugar, 1, 2, 3, 4, and 5. And I would like you to highlight using this 5 little apostrophe, that means prime. So that's something that could be really useful in your notes, just so you understand the way that DNA runs when we start talking about 5' to 3'. When we talk about DNA, we say that it's a double-stranded helix. That should be a good keyword to put on your cue cards as well. It's made up of 2 chains that run anti-parallel, and this is where our 5', 3' comes in. You can see we've got 5', 3', and then 5', 3'. So that's what it means, is it's anti-parallel. So yeah, they run exactly parallel to each other, but they're kind of flipped on their head. So anti-parallel is something that you should go and look up and define, and probably in your own words, and something you should have on your cue card. You can then see these little green, blue, red, and orange boxes, and the nucleotide base pairs are in there. So adenine always pairs with thymine, and guanine always pairs with cytosine. And I always remember that because the G is kind of round and the C is kind of round. So with our T and our A, we only draw straight lines. With these ones, we have to draw curved lines. So that's the way that I always remember that A goes to T, G goes to C. I just want to cast your mind back a little bit. If we're thinking about running a double-stranded helix, and these have to be parallel, which means they can't ebb and flow and cross over. If they're running parallel, we have to look at our bases as these purines and pyrimidines. So we just said A is to T and G is to C, and you can see that a purine always pairs with a pyrimidine. And that's another key thing that you might like to mention when you're talking about pairing of the bases. A purine pairs with a pyrimidine always, and guanine pairs with cytosine, adenine pairs with thymine. Because if you think about it, if you paired two pyrimidines together, the backbone would ebb and flow, it would go in and out and in and out, and it would be kind of curvy, look more like a caterpillar instead of that parallel, which is the word that we use to describe this. So because of the way that these bonds, one has to run in the opposite direction, and that's why we call it anti-parallel. So this means that the base strand of DNA dictates the second strand. So it creates kind of a replica, but in the exact mirror or opposite. So if we think about, if you know on the left-hand side, you've got a guanine, you know on the right-hand side, you've got a cytosine. So when we replicate DNA, we can only replicate one side and still have enough information from the other side. And that's more of some year 12 stuff that you start talking about with tRNA, mRNA, and rRNA. But it starts here, and by understanding that we can only copy one side, and that will be enough for us to understand. We also need to know that the reason that we call it a helix is because they spiral around an imaginary axis. And the way we call it double is because it has 1 and 2. So it's not just one spinning around, it's two spinning around, and they spin around together. We also think about Chargaff's rule. So in DNA, the proportions of A and T are pretty much equal, and the proportions of C and G are equal. This doesn't mean that A and C are equal or T and G are equal. What it means is if you ran through the code and you highlighted every single A on each side of the DNA, and then you highlighted all the T's, you get the exact same number of tick marks, or pretty close anyway. And this is the same for C and G because they pair. So this is what we talk about when we pair these base pairs. So if the order along the DNA chain is known, the sequence of bases on the second chain can be inferred. And you might be asked to do this in a test or an exam, and it's a really, really easy 2 marks. All you need to do is identify 5', 3', and then that it runs in the opposite direction, and you'll never be given a whole genome or a whole gene even. You'll just be given a short little section, and you just have to copy it down. So for example here, if we put a 5' here and a 3' on this side, we then put a 5' here and a 3' on this side because remember they're anti-parallel. You can then see that this base sequence dictates the second sequence. So we've got our base sequence along here, GGTA, CGTA. But if we replicate it straight underneath, T is to A is to T, C is to G, G is to C, and so on. And that's the way that we can dictate the second side of the helix. So double helical DNA consists of 2 nucleotide chains held together by weak hydrogen bonds between the complementary nucleotides. So these ones have weak hydrogen bonds in here. That means they can split and form back together. And they use that a lot when they go to replicate, when they go to synthesize in the S phase of interphase. Whereas the backbones along here, these phosphodiester bonds, they're really strong because they need to be held in order because these read like a code. So hydrogen bonds between the complementary nucleotides, but phosphodiester bonds along the backbone. What this means is we can actually heat up and dissociate and then resociate a DNA sequence. So the hydrogen bonds are easily broken with a low input of energy because they're quite weak. So if a solution of DNA is heated to only 90 degrees for 2 minutes, each double helical DNA molecule separates in a process called dissociation. They don't associate with each other anymore. And these form 2 single chains of DNA. This heating doesn't break the strong sugar phosphate bonds that join the nucleotides into one chain, so that's that phosphodiester bond down the backbone, and it doesn't break the phosphate from the sugar from the base, It just breaks the two bases apart. If this solution is then allowed to cool, the complementary regions of the chains pair back up and the hydrogen bonds reform. The DNA returns to a double-stranded helix, and this is called reassociation. So they're pulled apart, and kind of like magnets, they slip back together where they best fit. So pairing between complementary DNA chains or parts of the chains from different sources is referred to hybridization, and this is where we get into gene splicing and being able to design and code for different genes, and we'll come back to that a little bit later. So this is a picture of what that kind of looks like. So you've got on the left-hand side, you've got all of your double helical DNA. We put in some heat and energy. They dissociate, they cool, and then they reassociate because the little bonds in here are reforming again. So while that's all happening, we're talking about having DNA in a vial. The total length of DNA in a diploid human cell is more than 2 meters long. That's not, if it was small and then we blew it up and we scaled it up, that's actually how much DNA we have in one diploid cell. So one of our somatic cells. Yet a human cell is typically only about 30 micrometers long. Or 0.03 millimeters if you think about that in a diameter. So it seems strange or kind of impossible that there's this much DNA in one cell, let alone just the nucleus of that cell. The nucleus is even smaller again. So the width of DNA double helix is about 2 nanometers and tiny, tiny wide. But the DNA double helix becomes tightly coiled around proteins known as histones, and these coils become further coiled into supercoiled coils called nucleosomes. And these two bolded words are something that I would like you to put on your cue cards there. Histones and nucleosomes, and you might want to draw a picture of what they look like. So this coiling enables the DNA to be organized into chromosomes. So it starts to look a little bit like this. So we're within the nucleus, you can see our chromosomes, and you can see this tangle happening here, but what it is it's a ring, kind of like a spring, of these then further little nucleosomes, and then it coils around a histone, which is one of these tiny red balls. So a nucleosome, it's curling around here, but a histone is one of these tiny red balls. So this is kind of going in from super zoomed to not as zoomed. And so you can see that they've curled once, they've curled again, and this is because they want to increase the surface area. They want to increase the amount of information that they can put into one chromosome. So this is only in one of our chromosomes. This is the last little section for DNA structure. Transcription and translation is something that you'll do in units 3 and 4, but it's kind of always good to have a bigger picture of where you're going with this. So protein synthesis consists of 2 main stages, transcription and translation. The first stage is transcription and occurs in the nucleus of the cell. So there's a section of DNA, say along here, which contains a base sequence for the gene that's transcribed. So let's say this is the base sequence here in orange. So it's transcribed, it's copied, but in its anti-parallel, it's mirror. So it's copying a U because this is RNA. When I talked about uracil before, it's another one of your pyrimidines. It's smaller, and it copies it. And then it sends it out into the cytosol to be translated into proteins or amino acids to create proteins, which creates your phenotype. And we'll go through phenotype versus genotype in a little bit more depth a little bit later on. So each 3 of these, depending on where it starts, so it goes 1, 2, 3, you must need this one here. And there's 20 amino acids. So if you ever have like a pre-workout, or you're doing some proteins, or you have a Gatorade, it's got amino acids in it because what it is they're the building blocks for your protein synthesis. And that's the end of the DNA structure.